Hello my dear friends, welcome back to my YouTube channel. Myself, I am LAVKUSH JOGI. I have completed M.SC in Agriculture Seed Science and Technology, Specialization Seed Science and Technology from HNBGU Central University, SRINAGAR, Uttarakhand in 2020. I have got 52 rank in P.HD program Seed Science and Technology from Indian Council of Agricultural Research ICAR All India Entrance Examination for admission in 2021. Today I am going to tell about the topic Seed viability and vigor testing. Seed viability. Seed viability is an important component of seed quality. A seed is considered viable when it is live and potentially capable of germination. Hence, seed viability refers to the capability of a seed to germinate and produce of a new plant. It is impossible to determine whether a seed is live or dead from external physical appearances, wise seed shape and color of seed. Hence, viability testing is used to determine the percentage of viable seed in a given lot. Seed viability is one of the indications of potential performance in the field. The germination test and Tetrazolium test are two important viability tests used by seed laboratories. Why does seed lose viability? Adverse weather conditions during seed development and maturations, drought, excess water, extreme temperature etc. Nutrient deficiencies and pesticides injury during seed development and maturations. Environmental condition after physiological maturity during harvesting, drying, cleaning, storage and handling of seeds. Different method of testing seed viability. Tetrazolium test (TJ), indigo carmine test, urine sugar analysis paper test, radiographic soft X-rays test, embryo excision test, glutamic acid dehydroxylase acidity GADA test, ferric chloride FC test, seed leaf conductivity test LC, leaf color LC test, seed crushing test, accelerate aging test. Rapid assessment of viability. Tetrazolium test. In addition to germination test, Tetrazolium test is most widely used to measure seed viability. It is a biochemical test developed by a German scientist Georg Lacan in 1942. Distinguishes between the viable and dead tissues of seed embryo on the basis of dehydrogenase enzyme respiration enzyme activity. Upon hydration, the activity of dehydrogenase enzyme increased, resulting in release of hydrogen ion H+, which reduce the light yellow or cream colored compound tetrazolium salt solution 2,3,5 triphenyl tetrazolium chloride into red color called pharmazone. Hence, it is used to differentiate between metabolically active and inactive tissues. For pharmazone stains living cell red while dead cells remain colorless. The viability of seed is interpreted according to the staining pattern of seed. Methodology seed hydration. By soaking seed in water for a specific period of time depending upon the species. This is done to active hydraulic enzyme hydrogenase and stimulate respiration. Cutting or puncturing of seed, puncturing seeds. This is done to allow and penetrations of tetrazolium solution into internal tissues. Staining of seed. It is done by soaking seed in tetrazolium solution for a specific period of time to allow staining of viable tissues in the seed. Tetrazolium is used direct 0.1 or 0.5 or 1.0% solution at 33 to 35 degrees Celsius temperature for 24 or 48 heart pH of 6 to 8. Evaluations of seed evaluated according to staining pattern. For detailed producers in interpretations of result, consult AOSA Tetrazolium Testing Handbook 2000. Preconditioning of seeds before tetrazolium test. Seed directly placed in tetrazolium solution in case of pea and beans. Bisect longitudinally before placing in tetrazolium solution cereals and grasses. Glumules of seed may be removed before testing EZ08. The seed may be subjected or packings blue. The embryo or they may be bisected laterally. Very small seed of grasses. The seed coat may be removed cucurbits. The seed coat may be secreted above the embryo luteus. A piece of the embryo may be excised and stained. Example, pines. The accuracy of the tetrazolium test requires a sound knowledge of the seed and seedling structure and understanding of the mechanism of the reactions leading to the staining of the seed. Ability to interpret the staining pattern or critically examining the seed topography. Experience gained through keep interest and natural skills. Advantage: seed viability can be estimated in a much shorter time compared to germination test. It is used for dormant seed in the combinations with germination test. Useful test where seedling growth is extremely slow, particularly is in some medicinal plants. Test required a close examinations of seed, and thus seed analyst can often get an idea of the reasons for poor viability of seeds. Since dicot seed need not be damaged during tetrazolium test, the same seeds could be used for further investigations. Disadvantage: relations of staining pattern and their interpretations in terms of viability is difficult and required considerable experience. Distinctions between normal and abnormal seedling is difficult. The total work included in the test requires greater number of man hours than germination test. This test alone is unable to differentiate with dormant and non-dormant seed. Microorganisms harmful for seedling emergence remain undetected. Correct evaluations is possible only after prolonged experience. Seed vigor. It is an important seed quality parameter which need to be assessed to supplement germination and viability test to gain insight into the performance of seed in the field or storage. Seed viability is the state of being live, whereas seed vigor denotes degree of liveness. Star Congress in 1977 adopted the following definitions of seed vigor. It is the sum total of all those properties of seed which determine in potential level of performance and activity of non-dormant seed or seed load during germination and seedling emergence. AOSA, Association of Official Seed Analysts, defined as seed vigor The sum total of all those properties of seed which upon planting result in rapid and uniform production of healthy seedling under a wide range of environment including both favorable and stress conditions. History. Walter and Insen, 1911 German pathologist, give the original idea of seed vigor based on their work on cereal seed are infected by Fusarium species. The concept remained obligous still. Isle 1957 defined on the basis of his experiment. His defined seed vigor as sum total of all those attributes which favor stand displacement under unfavorable field conditions. The major drawback of this definitions was that he gave more emphasis on environmental than seed itself. Importance of seed vigor. Crop performance, which is a interactions between GX, is greatly influenced by seed vigor. Good and unformed crop stand of genetically pure variety are crucial for exploiting of full genetic potential of a improved genotypes. Low vigor seed result in suboptimum plant population and are plant growth resulting in poor yield in a otherwise improved and high yielding cultivar. Vigor difference are relatively more important in case of expensive seeds such a hybrid seed of vegetable crop where the production system demand unformed crops tend to obtain optimum quantity and uniformity in crop produce. Vigor of seed is most important in protected cultivation where required 100% uniformity in plants. Factor influencing seed vigor. Seed vigor is the quantitative character controlled by a number of factors affecting performance of seed and seedling. These factors are genetic constitutions, environment and nutrition of mother plant, seed maturity and harvest, seed weight and size, mechanical integrity of seed, duration and aging, pathogen, pest and attack. Criteria for selection of a seed vigor testing method. Two different criteria have been employed by STA, International Association Seed Testing. Seed vigor committee to evaluate of performance of seed vigor test method for different crops. First, reproducibility of vigor method. Second, relationship between vigor test result and seedling emergence in the soil, in field soil. There are no universally accepted vigor test for all kind of seeds. Differentiate test for estimation of seed vigor. Physical test: seed size 100 to 1000, seed weight, physical soundness of seed, soft X-rays. Performance of growth over physical test. First count: speed of germination, seedling growth rate dark, seedling dry weight, seedling vigor index, length and mass. Shred test: first cold test, second bricks gravel test, third paper piercings test, for compact soil test, fifth wet or dry soil test, sixth pathogen infected soil test, seven accelerated aging test at controlled deterioration test, 9 low and high pH test, 10 water sensitive test, 11 osmotic state test, 12 oxygen state test. Biochemical test: Tetrazolium test, seed lithic seed or electrical conductivity test. Physical test: seed size 100 or 1000, seed weight, test weight, physical soundness of seed, soft X-ray test. Growth test. Principal tests are based on principle that high vigor seed grow at faster rate than low vigor seed. Even under favorable environment conditions, high vigor seed rapidly germinate and establish in the field. Therefore, may be method used to determine the rapidity of growth of the seedling will give an indications of seed vigor level. These include first count, speed of germination, seedling growth rate dark, seedling dry weight, seedling vigor index. First count, it is done along with the regular germination test. In this, number of normal seedling germinate on the first count day as specified in the germination test for each species are counted. Higher the number of normal seedling, greater is the seed vigor. Seedling growth rate and dry weight. The seedling are grown either in laboratory, ground, greenhouse or field. In laboratory, controller paper towel method should be followed. The randomly selected seed are planted in the center of the moist towel paper in such a way that the micropeals are oriented towards bottom to avoid root twisting. The rolled towel paper are kept in the germinator at the temperature recommended for crop in reference. After a specified period of time, 5 to 10 days, 12 paper are removed and 5 seedling are selected. Then length root plus shoot is measured and mean seedling length is calculated. Seed lot producing, the taller seedling are considered more vigorous than the seed lot producing shorter seedling. For dry weight determinations, the seed are removed and dried in a hot air oven 100 degrees Celsius for 24 hours. The seedling dry weight provide additional information for assessing seed vigor. Speed of germination. 100 seed is in for replication are planted in recommended substratum for germination. Then they are kept in a germinator maintained or recommended temperature for the crop in reference. Number of seedling emerging daily are counted from day of planting the seeds in the medium till the time germination is complete and germination index is computed as seed lot having higher crop germination index is considered become more various. Day count 1 2 3 4 5 6 7. Number of seedling emerged lot A 0 0 0 4 3 20 0. Number of seedling emerged lot B 0 0 15 50 25 0. Seed vigor index SVI. This is the calculated by determining the germination percentage and seedling length or dry weight of the same seed lot. 100 seed is the 4 replications are germinated as prescribed for the crop specifying germination test, while evaluating the number of normally seedling at the time of final count of seedling length of 5 randomly selected seedling are measured and then average worked out. Similarly, these seedlings are kept in hot air oven in 100 degrees Celsius for 24 hours. Find out the dry weight of the seedlings. Seed vigor index is calculated by the multiplying germination percentage and seedlings length dry moisture dry weight as follow. SVI length germination equal germination percentage multiply seedling length centimeter. SVA mass equal germination percentage multiply seedling dry wet milligram. The seed loss showing the higher seed vigor index is considered to be more vigorous. Kharand Vale 2011. Stress test. Cold test. This test was the originally developed for corn. The ISLE 1950 is a USA but can also be used for other crops like cotton, barley and soybean and other summer vegetables. In USA, the corn is planted in spring when soil is cold and humid and weak seed do not germinate and establish principle. Weak seed do not germinate in cold and humid soil, whereas vigorous seed are capable of germination under such situations. Grid and to sand soil fill it into a tray upon 2 cm depth. Place 50 seed over stand soil. Cover with another 2 cm layer of soils. Soil compacted and water in added to make the soil about 70% of its holding capacity. Its water holding capacity. Over with polythene. Place in germinator at 10 degrees Celsius for 7 days. After 7 days, put them in germinator at 25 degrees Celsius and seedling emerge after 4 days are counted higher. The germination greater is a Vigor. Brick gravel test. Hitler test. Hitler 1917 developed this test. From the experiment we concluded that seed are still affected with the fusarium were able to germinate in a regular test but could not emerge from brick gravel of 2 to 3 mm size. However, healthy seed were able to emerge bricks gravel principle based on principle that weak seedling are not able to germinate enough force of overcome the pressures of bricks gravels. Perry 1984 established that the method is reproducible and associated with field emergence in cases with wheat and other cereal crops. So fill it in germination box. Try leaving 3 cm impurity to top. Place 100 seed in each box tray. Speed to 2.5 cm layer of proteus bricks gravel 2-3 mm in size over the seeds. Cap box in germinator and at appropriate temperature after prescribed period. Remove germination box. Work out percentage emerge seedling. Paper piercing test. The test was suggested Fritz 1965 principle similar to brick gravel test. Higher vigor seed are expected to produce strong seedling which can prices particular type of paper, while seedling of poor vigor seed may be not able to pierce the paper. Quality of paper. Basic weight 9 gram m square. Thickness 0.4 mm. Dry distributing strength 0.3 kg oblique centimeter square. Breaking length 1000 to 5000 mm. Filtering speed 500 ml in minute. Weight versus bursting strength 150 mm. S content 0.1%. Fiber compositions, chemical wood pump with high alpha percentage. Place seed on the 1.5 centimeter moist sand in a tray. Cover with special paper. Cover it 2 centimeter moist stand. Keep in germinator at 20 degrees Celsius for 8 days. Count the seedling emerged. The test of highly dependent on the quality of paper of mentioned. This test should only be used for testing vigor and such quality of paper is available. Accelerated aging test AAT. Accelerated aging test. The test was developed by seed testing lab Mississippi State University, USA, determining the storage potential of seed lots. Principle: the aging process is accelerated by subjecting seed are high temperature 40 to 45 degrees Celsius and high relative humidity greater than 90%. The seed lot which show high germination in AAT are vigorous and expected to maintain high viability and abundant storage. 100 seed or 4 replications are dried in a fine muslin cloth. Fill the lower portions of the jar with water. Ensure that seed do not touch the water. Cover the jar with lid and seal them airtight. Place the jar or accelerating aging chamber or 45 plus 2 degrees Celsius temperature for 3 to 5 days. Remove the seed and cool them. The seed by using normal germination test. Higher the percentage germination, greater the vigor and seed load. Biochemical test. Electrical conductivity test. Principle: weakening of the cell membrane in poor vigor cell causes more linkage of water soluble compounds like sugar, amino acid, electrolytes, etc. when immersed in a water as a compared high vigor seed having intact cell membrane. The measurement of electrical conductivity of the leads via conductivity meter gives as an accurate estimation of membrane permeability. The value of this test appearance of the restricted to the large seed species of the family Leguminaceae. Procedures: weight to 5 gram seed sample, surface sterilize with 0.1% HgCl2 for 5 to 10 minutes, wash with distilled water thoroughly, immerse seed in distilled water 100 ml at 25 plus 1 degree Celsius for 10 to 12 hours. Remove seed with clean force. Water left is known as leachate. Warm electrical conductivity meter for 30 minutes. First measure to conductance of distilled water in Vigor. Cleanse electrode with tissue paper and measure the conductance of lead knob. Actual EC of leads, EC distilled water. Lower the EC volumes, higher the vigor of seeds. Conclusions and further roles. Seed vigor testing is an important component of seed quality analysis, but still this information is neither required nor displayed on seed level. This is due to the fact that there is no universally accepted and perfect method of testing seed vigor of the particular crop. Hence, research is needed of further refine the existing methods and to develop new method which could be universally accepted.